When we think about a fungus, we usually associate it with something that's negative, right? We usually think of fungal infections, mildew, or mold of growing over our food. Yet here I am showing a cartoon of fungi shaking the hands of a tree. Today I'm going to talk about my research, which tries to find out how mycorrhizal fungi, which are growth-promoting fungi, improve the nutrient uptake in the soil for plants to grow. So to prepare you for my presentation, I will first highlight the outline of my talk. I will tell you why it's important to study this type of relationship. I will then mention two very important terms, mycorrhizae and rhizosphere. This is where you have to pay very close attention because there will be a quiz at the end. No quiz. I'll then mention the method that we use, including the experimental model that was designed specifically for this. I'll show some results, particularly how we're able to detect nitrate in the rhizosphere, and then finally I'll mention some of our work that we plan to do in the future. So it's estimated that in the year 2050, the world's population will increase by about 9 billion. During that time, it's estimated that there will be a 60% increase in the demand for food. Now, thinking about these estimates, and thinking about our current global challenges, for example, climate change, trying to grow food crops and biofuel crops sustainably, and trying to produce it in a way that provides sustainable food security and quality, that means that it will become increasingly difficult to meet the world's demand at a low environmental cost. Having realized these concerns, it is now more prevalent to invest in soil research now more than ever. What we do know is that more of the same agricultural approaches will not work. They've led to a lot of leaching of nutrients in water treatments, especially when excess fertilizer has been used, and it's caused a loss of biodiversity in our soils. Right now, we need to focus on using alternative approaches for agricultural purposes to grow crops in a very sustainable manner. One way to do that is by using what you call mycorrhizal fungi. They're a type of growth-promoting organism that improves the nutrient availability in the soil for plants to grow. Mycorrhizal fungi can be used as a chemical fertilizer it could be used as an organic fertilizer instead of a chemical fertilizer while providing the same benefits. So, term number one, mycorrhizae, what does it mean? So, a mycorrhiza, plural mycorrhizae, is the symbiotic union between a fungus and a plant species. During that relationship, a fungus colonizes a plant's root. It forms a covering over the root, or what you call a fungal sheath or mantle. From that mantle, the fungus grows thread-like extensions that extends into the soil. So it becomes an extension of the plant system. By doing that, it's able to penetrate a wider surface area in the soil, absorb nutrients, and bring it back for the plant so that the plant could use it. The point I'd like to note here is that this type of relationship can be used on an industrial scale to grow plants sustainably. The area that we're most interested in looking at in the soil is called the rhizosphere, term number two. The rhizosphere is the area around the plant root that's most actively involved in nutrient uptake processes. It contains the richest composition of nutrients and microorganisms that are present in the soil to help the plant grow. Now, in this type of relationship, there are several events that are happening to cause this relationship to continue. One, there are several nutrient pathways that exist that help to shuttle nutrients from the soil to the fungus and to the plant. In these types of nutrient pathways, there are transporter and sensor proteins that exist, but they're not very well understood. Another event that occurs is the distribution of nutrients, or I should say the redistribution of nutrients that facilitates that type of relationship. My project aims to understand how these nutrients get distributed in this type of relationship. It has been challenging to study this nutrient distribution in the rhizosphere, mostly because the techniques that have been used have lacked a way to reliably detect what's going on in situ on a sub-millimeter scale, and they've led to a lot of bulk averaging of nutrients, making it incapable to understand what's going on different spatial scales. To overcome these challenges, we decided to use infrared imaging. Infrared imaging is a technique that allows you to get a chemical image of your sample of interest. By shining infrared light on your sample, you're able to get a spectrum that shows the composition of what you're looking at based on the different molecular vibrations that are present in your sample. Because you're collecting several spectra over a region, you're able to see the variation that occurs as you integrate one area in your spectrum over another. So I'll give you an example. So here I'm showing the cross-section of a poplar stem. The bottom area represents a visible image of what the cross-section looks like. The area in the red box is the region that the chemical image or the infrared image was collected over. Up here is a representative average spectrum of what you might get at one of the pixels in this red box. The different images here, which are the chemical images, they show the variation in different components. So for example, aromatic compounds, which end up here at about 3250 wave numbers. It shows you how proteins are distributed, as well as lignin and other compounds. What's advantageous about this method is that it's non-destructive. It allows you to get a spectrum at each pixel that you're looking at, and it's sensitive to simultaneous changes that are occurring. What's even more important is that it provides highly specialized resolved chemical information that determines where specific events are happening, especially using a synchrotron source. The other advantage of using this method to look at the experimental model system that we're dealing with is that our nutrients of interest, our phosphates, our nitrates, and our sulfates, have non-overlapping peaks that should make it easy to resolve once we get our data. But like any other method, it does come with its own challenges, right? And so some of the things that we had to consider when dealing with this method is, one, what type of substrate should we grow the plants on? So we needed to come up with something that was infrared compatible, something that was cost effective, and something that would also allow for good plant viability. In addition to that, we needed to figure out what mode of spectroscopy that we should use, whether it was transmission or reflection, which inherently determines how thick of a sample that you could get. That and a couple of other challenges we all had to think of, but most importantly, we had to come up with something that was really reproducible and reliable. So this is the experimental model system that we came up with. It entails dipping an infrared compatible slide, a cold slide, into the hot nutrient media. Once you've done that, it forms a thin coating of the nutrient gel over this infrared slide. From there, a poplar seedling is embedded into the slide, and it's allowed to grow in it for about 8 weeks. Once it's grown to its full size, we then dry it down before we get infrared images from it. What's unique about this system is that it allows you to control the different concentrations in the media. So it contains vitamins and nutrients that the plant would get if it were outside in the regular environment. In addition to that, it allows you to add microorganisms that you're interested in studying and watch the desired effects. But before we could collect our chemical images, we had to figure out what nutrients in our media came from the different peaks in the spectra that we collected. So we decided to first look at nitrate. So why nitrate? It's one of the most essential nutrients to the plant, and it's needed for the plant growth, for plant and root, for shoot formation, leaf formation, and for the production of proteins that are essential for biochemical and biophysical processes that the plant needs to carry out. So one of the main sources of nitrate in the media is ammonium nitrate. And to figure out where the nitrate peak or the ammonium peak was coming from in our spectra, we used isotopes of ammonium and isotopes of nitrate. If you look over here, you'll see that there's a change in the wave number frequency from 1340 to 1305 that we've attributed to the nitrate stretch. Similarly, by using the ammonium isotope, you could see a change in the wave number from 1435 to 1425, which we've attributed to the ammonium nitrate. So we now know where to look for nitrate and where to look for ammonium in the spectra that we get. Once we did that, we needed to find out a way to quantitate the absorbance values that we were getting. So how could we determine the nitrate concentration from the spectra that we get? So we did a series of nitrate in different concentrations of nitrates in solution from 7 millimolar to 20 millimolar. From there, we were able to read off the absorbance value of nitrate at 1340, which we found out from the previous slide, and plot that absorbance value versus the nitrate concentration to yield this equation that we could then use to determine the concentration of nitrate at every pixel from the chemical images that we obtained. So about the chemical images. On here, I'm showing data from a system that includes only the plant. The top image shows the visible image of the plant only over the glass slides. The thin thread-like extensions that you see are the root hairs of the plant. The bottom image shows the corresponding chemical image, and the spectrum over here shows the concentration of nitrate versus the distance taken from this region over here. So what does it show? It shows that there's a nitrate gradient that exists around 400 microns away from the root. This gradient range is a factor of 2, and that we could also see after 1000 microns, there is no significant change in the nitrate concentration. What this implies, and is not surprising, is that the root hairs, which are about 800 to 1000 microns in length, are only effective up until that point. When we look at the system with the plant and the fungus, so again you have the corresponding image of the plant. The brown spots that you see are fungal colonies that have grown over the plant. The bottom image represents the corresponding chemical image, and again this line plot represents the nitrate concentration versus distance extracted from this region over here. So what do we see? There's a nitrate depletion region that exists about 1000 microns away from the plant roots. There's a huge nitrate gradient that's about 30 times in range, and a decrease in nitrate concentration that begins to occur after 2000 microns. This shows that as the fungal colonies decrease in population, the nitrate concentration decrease, and you'll see why in a minute. When we plot the two of them on the same graph, so the orange represents plant only, and the blue one represents the plant and the fungi, you could see the difference in intensity of the nitrate that's available once the fungus is added to the system. We see that there's a higher nitrate concentration all throughout the system and even beyond 3000 microns away from the plant. Over here, the highest nitrate concentration is about 4 times the amount of the control concentration that's actually in the media. This here shows that the fungus plays a key role in improving the nitrate availability for the plant. So, looking at nitrate and sucrose, which is a carbon source in the media, we first see that they're both highly variable. So the panel in B represents the nitrate distribution, C represents the sucrose distribution, and the two plots at the bottom represents the nitrate versus the sucrose concentration. What it shows is that in areas where you have the highest nitrate concentration and the highest nitrate gradient is the areas that correspond to the lowest sucrose concentration. So we see this inverse relationship. The areas where you have the fungi, you have the highest amount of nitrate, which means that it's pulling it up or sequestering it from the soil for the fungus to use. In return, it is consuming the sucrose as a food source. Right? This plot here really shows the symbiotic sharing of nitrate and carbon that's present in the system. And I think this model really highlights how we could use it to predict changes in the nutrient distribution in the rhizosphere so that we could use it to grow plants. In summary, we've created an infrared compatible method to actually grow plants and image the nutrient distribution that's available. This system can be extended to include other microorganisms, for example, growth-promoting bacteria. In addition, we've been able to show that there is symbiotic sharing of nitrogen and carbon sources using this method. Right now, we're trying to determine if the fungus has a preference for the type of nitrogen that it's using, whether it's ammonium or nitrate. In addition, we're observing the changes in the nutrient distribution for plants grown under different nutrient conditions. Under low nitrate conditions, which would represent marginal soils or poor nutrient soils, as well as adding additional microorganisms, for example, growth-promoting bacteria, as well as the mycorrhizal fungi. In the future, we'd like to use multivariate analysis to determine, to build a more robust library of all components and hopefully observe the nutrients in real time using wet samples as opposed to drying them down. So the work that I've shown today would not be possible without the financial support from the Department of Energy, as well as Stony Brook's Chemical Biology Training Program. In addition, I'd like to thank my collaborator, Dr. Leland Check, who's been instrumental in growing the plants, my group members, especially Randy and Vito, for the technical expertise and their support, and my advisor, Dr. Lisa Miller, for her continuous support, her guidance, and her encouragement. With that, I could take any questions that you have.
Okay, we have anybody have any questions? Yes.
I was wondering if the fungi that you use are specific, or is it any type of fungi that is effective in this?
So different plants form this type of association with specific types of fungi. So this one is one that's widely found over a wide range of species. So in particular, it will form this relationship with the poplar plant, which is what I use here. So people do that. There are a lot of small startup companies that are now introducing mycorrhizal fungi into food crops to grow food crops, and I'm sure tomato is one of them. But when the pictures that, so on here I don't have any pictures because they're all dried, but those that are grown with the fungus, especially in the low nutrient conditions, you do see a big difference in the biomass that's formed. Okay.
What makes it difficult to do it well?
So water has this huge absorbance peak that covers some of the areas of interest that we're looking at, especially the proteins.
The biomass, there is an increase. This is in the root biomass, in the sense that can you consider it when you're growing plants in drought conditions? Do you think this would help?
Yes. So for sure, with this type of relationship, it helps forage a wider surface area. So even if you were to add a small amount of chemical fertilizer or a small amount of water, it would definitely improve the type of relationship and have more biomass.
Okay. Okay, thank you, Victor.